good afternoon. 12:41 p.m. Eastern Standard Time. 12:41 on Tuesday, December 5th, 2023. My hair isn't as bad as I thought it was going to be today. Little George Rafferty. And what's fascinating about the particular date, Tuesday, December 5th, 2023, in the context of this show, is that this is a redo, if you will. This is a redo, a remake of a video that we did in November of 2021. Just turn this up just a little bit here, okay? And the content is, it's just a reading with some narration of an article that was in Endpoints News, that was ran on August 25th of 2021. So when I first read this article in November of 2021, it was 3 months after it came out. And that it was fascinating how much this article titled Inside the Quixotic 30-Year Quest that gave us the COVID-19 vaccine treatments, vaccines, and could unlock vaccinology's new holy grail. And I'll show it on the screen here. And you can just, the original article can be found on ENDPTS. ENDPTS.com stands for, presumably, Endpoints News. And I'm not familiar with Endpoints News and their various affiliations. Actually, I need to scroll back up here. One picture of it is the 30-year hunt for the perfect antibody. And I think one mistake that I made when I first read it was I didn't realize that the real headline of the article is the 30-year hunt for the perfect antibody. What I read, I thought this was just a picture, like spicing it up, if you will. But I thought that this, which is the subtitle, was the real headline. The subtitle reads, Inside the Quixotic 30-Year Quest that gave us the COVID-19 treatments, vaccines, and could unlock vaccinology's new holy grail. And this was from August 25th, 2021, and updated September 28th, 2021. Presumably the same year. It doesn't state the year, but presumably 1 month afterwards. Written by Jason Mast. Okay. I read this article because it mentions several people who are related within the United States, or I should say not related, but who were instrumental in the creation of and the operation of something called the VRC, the Vaccine Research Center, a new division that was started within the United States National Institutes of Health, specifically Anthony Fauci's National Institute of Allergies and Infectious Diseases, or NIAID, in 1997 or 98. It was a whole new division within NIAID. It's odd. I've mentioned this before. I mean, the whole division seems to focus on vaccines anyways, and now, we need a division to focus on vaccines. Like McDonald's says, we just need a new division that focuses on nothing but hamburgers and French fries because there's just too much other stuff on our plate. It's okay, whatever. Anyways, so I read this 2 years ago, and here I am. I'm going to read it a third time, basically. I read it once over 2 years ago. I didn't like the way it came out. It was just a little choppy. I didn't make it through, I think, to the finish. So then I tried it a second time with a whole lot of new additional information. And now 2 years later, we have even more insights. We've learned more about companies that are mentioned in this article, such as Modex, M-O-D-E-X. We've learned the importance of John Mascola being a key person, science officer now at Modex, and how odd, and we appreciate what Modex is doing with respect to a future vaccine antibody technologies where they are going to be using transfection to create antibodies directly in people. No more of this creating the antigen nonsense and then the body makes the antibodies to that. This is well understood if you take the time, listen to the antibody patent paradox, and understand that what had become the largest sector, I believe, of pharma, which was monoclonal antibodies, had a major underlying fundamental patent issue or paradox. And they are going to get around it by having people make their own antibodies. And they're going to lower the cost of manufacture because vaccines can have a limit on how dirty they can be relative to monoclonal antibody therapies, which have to be as pure as can be. The main reason why they cost so much is because of the purification. Most people haven't understand that. So guess what? You don't have to purify, I guess, as much the mRNA or the DNA that's injected in shots, as long as they're considered a vaccine. You can save a ton of money. No more spending money of days and weeks and months of purification. Just straight to market. Redefine everything that could go wrong in a human as a vaccine preventable or minimizing disease. Everything. And this is exactly what's happening. And the other reason why this article I just found incredible is because one of the key people mentioned in this article is Nancy Hagwood, Higwood, the very person of all the 330 million Americans that went to the FBI in 2007 or 8 and said, you know, Bruce Ivins always made me a little uncomfortable. He could definitely be the lone wolf responsible for the anthrax letters. Case closed. It's all Bruce Ivins. As minimal as the evidence that Hagwood brought forward is, okay, I might give her a little bit more benefit of the doubt. He said, she says type thing. If it wasn't for the fact that she is so instrumental and involved in the biggest event potentially in our lifetimes. How is it that you can be the sole witness of Ivan's instability, the guy responsible for the anthrax letters, supposedly, and which has led to all of this biomedical pharma industrial complex, erosion of constitutions, the creation of BARDA, actually, etc., etc., etc. And then it just happened to be in a select group of 20 or 30 people who are at the forefront of all of the new fascinating technology, which is being unleashed upon the world by scaring people about a novel, deadly, spreading pathogen. It's just hard to believe. It would be almost as crazy as if Anthony Fauci himself was the person that said, Ivans emailed me about my college fraternity and made me uncomfortable. It's almost that bad. And now, although this article doesn't even mention that Hagwood was that person, it is also shocking. And I can't get this message out enough, which is why I'm so heavily censored, that Hagwood has known Malone since 1989, presumably, based upon metadata that I've been able to attain from the Big Shot AIDS vaccine book, which was published the week after September 11th, 2001. Meaning that the current truth, medical freedom, transparency, eagle movement, whatever you want to call it, out of the 20-something or so people you have on the stage, you have the initial primary suspect, Stephen Hatfill. You have Robert and Jill Malone, who must have knowledge about who really sent these letters and have all kinds of involvement with respect to let's just say, vaccine research out of the military right around that time, even working at the Winber lab, setting it up, matter of fact, near Shanksville. And Merrill Nass, who is the person that said the old anthrax vaccines are bad, a problem which can only be solved only by embracing, ta-da, new vaccine technology. I don't know if Merrill knows that or not, but she darn well should. To her credit, Merrill Nass does say that the threat of anthrax is remarkably overblown. And that's true. Okay? That's 100% true. Thank goodness that she says that. However, the dilemma of the madness of the vaccines is the pursuit to the ways that people have pursued solving that within the Beltway, if you will, is new vaccine technology. And the badder the old anthrax vaccines are, the more visible those injuries are in the U.S. military, the more the new vaccines will be heralded, praised, and ushered in and supported. Anyways, we keep learning more and more about the backstory of the individuals in this article, which is why I'm reading it a third time. And I'm probably going to do this with other videos as well. I may delete the old one just to steer people this way. I didn't say anything that I regret. It's just that we just keep learning more. So with that said, the old reading of this took almost 2 hours. I didn't do a breakdown of how much that was just narration or so on. I'm already 13 minutes in, as you can see. I don't have a chat window open up. I'm not doing chat today. If you send any super chats, I deeply appreciate it. I'm just incapable of showing the thanks that I should be showing right now. I'm just keeping it all business. And again, as many people know, life is taxing, but everyone's got a story. It's not that I'm unique in that sense. So without any further, but if you do, thank you very, very much for helping to support the program. Okay, so with that said, let us just get down into the reading. And by the way, you're going to have to sign up and pay for something if you want to go to Endpoints. Keep this a secret, just between you and me. You can go to my Houstonic.live or HoustonicITS.com, click on the M's, go to Mascola, and I have a full copy of the article there. And that's what I'm going to be reading from. So let me just scoot out of the side a little bit. And yes, it's amazing. 2 years later, and I probably only have 100 more subscribers. It's remarkable. And that's actually, maybe I have 200 more. But we just keep learning more. I was 48 when I read this article last time. Now I'm 50. It's just amazing. And we still are struggling to get this more people to see this message. But it's getting out there little by little. I know it is. I just have to be slow and steady, apply that slow and steady pressure, so to say. And that is what we do on the interdisciplinary Bright Web. Okay. Inside the quixotic 30-year quest that gave us the COVID-19 treatments, vaccines, and could unlock vaccinology's new holy grail by Jason Mast. So, John Mascola, America's soft-spoken, no-nonsense chief vaccinologist. Chief vaccinologist. Everyone thinks that it's Birx or it's Fauci or all the... No, Mascola is considered to be America's chief vaccinologist. I don't know what kind of a title that is, but just showing how many important people are not currently being discussed at all of these meetings and on within chats and on Twitter and so on. I myself haven't even really dug that much into Mascola. John Mascola, America's soft-spoken, no-nonsense chief vaccinologist, spent the year 2020 on the 4th floor of the Vaccine Research Center in Bethesda, Maryland, primarily working on 3 projects. There's a decent chance one of them has already been injected into your arm. Twice. Another was infused into hundreds of thousands of COVID-19 patients, potentially protecting them from hospitalization. And the third is a scientific codex, C-O-D-E-X, that bedeviled Mascola, his colleagues, and researchers around the world for the better part of 3 decades. That is an interesting word. So he's been working on 3 projects. The vaccines, presumably that's things like the Moderna and Pfizer. The second one is what's been injected into people to reduce hospitalization. I'm going to assume those are monoclonal antibodies of sorts. And the third is a scientific codex. And I suspect that codex somehow is going to connect to his company that he joined as now like the 4th main employee, the first being the previous NIH director, the name escapes me right now. How could I do it? Zarhuni, and the other being the married couple, Elizabeth Nabel and Gary Nabel, who go back to Gary Nabel in particular, the Vical Corporation connections, that DNA vaccine company where Robert Malone had his patents. Oh, my goodness. And I think this, so they have a company, Modex, which is going to be, as we've just learned, making monoclonal and making antibodies directly in people with transfection. Because again, it is our hypothesis, and it's no longer just speculation, it's a hypothesis right now, that the reason why there was all this noise about the badness of the spike protein and the reason why they chose the spike protein in the transfection was because they knew there would be problems, mistakes, illnesses, and deaths. But they just still wanted a way to be able to say, factor a bunch of things out and then say there was contamination and some accidents and some batch problems and whatever. To say, well, you know, at least the base technology works. We're going to use that next time just to make the antibodies. Sweet little trick that they pulled on everyone. Now you can see the boosting of this current New Zealand data story is probably going to be somehow connected to this. I don't know. Anyways, and the third is a scientific codex that has bedeviled Mascola, his colleagues, and researchers around the world for the better part of 3 decades. Before COVID-19, the largest and longest-running mystery in infectious disease was how to build a vaccine against HIV, the famously evasive virus that has claimed the lives of 36 million people since the 1980s. Within a year of the novel coronavirus's discovery, 2 vaccines were already available and shown to be 95% effective. Narrator's note, 95% effective doesn't mean 95% survive a challenge test. Okay? Doesn't mean every 20 people who were injected with the supposed SARS-CoV-2 virus survived and only 1 died. It means 95% of the people who were injected with things showed some signs of having overall improved effective protection. It's sort of like the MyPillow guy walking up to 20 cars and throwing a pillow into 19 out of the 20 cars. And he could say, you know what? I just provided 95% effective protection with nothing more than pillows. It doesn't mean he's going to improve the chances that any one of those people are going to live a crash. It just means that's what 95% effective means. End of narrator note rant. 38 years after HIV's discovery, you could fill encyclopedias with all scientists have learned about the pathogen, but it's hard to say whether or not we're any closer to a vaccine. Part of that disparity has to do with the coronavirus's comparative simplicity. Michael Farzan, who's an immunologist, last year put it, I guess in 2020, quote, "This is a stupid easy virus to vaccinate against." Stupid easy. HIV, unquote, HIV is, by any definition, the hardest. But part of it also has to do with all the tools researchers developed trying to inoculate against HIV, confronted by a microbial enemy more cunning than any humanity has ever discovered or encountered. The world's leading researchers were forced to go back to the drawing board. Crossing disciplines and continents, they came up with new tools to understand both viruses and the immune system, and new ways of turning that knowledge into walls and weapons against any pathogen, however stupid or ingenious it may be. Those same researchers jumped out in the winter of 2020 to study the coronavirus, unleashing tools honed on a far more cunning foe. Mascola, who had spent 30 years on HIV vaccine development, most notably launched the effort to find the first antibody treatment later marketed by Eli Lilly. Legendary AIDS researcher David Daiho, D-A-I-H-O, worked on his own antibody treatment and conducted crucial studies on whether these therapies were evading the virus. Neil Pate, P-A-D-T-E, said, quote, HIV was such a challenging virus that we had to come up with newer and newer technologies. So when SARS-CoV-2 broke out, we had a toolbox at the ready, unquote. He was a longtime collaborator of Dr. David Daiho. Daiho. I don't know how to pronounce it. I apologize. The timing, if this can ever said to be of a pandemic, was fortuitous. Before COVID-19, Mascola and other researchers were finally nearing, well, something. It's not quite a vaccine, though it may yet lead to one. Instead, it could turn into one of the best consolation prizes in the history of medicine, a byproduct of the vaccine hunt that may ultimately prove nearly as useful, a set of ultra-rare Y-shaped molecules that, at least in animals, could neutralize most strains of HIV, potentially protecting at-risk people for months or a year and offering a path to new treatments. The story of how researchers arrived at these so-called broadly neutralizing antibodies over the last 30 years is one of the hidden stories of everything that went right about America's pandemic response. The silent engine behind the vaccine and particularly the most effective treatments, antibody therapies from Vir, V-I-R, Regeneron, and Eli Lilly. Narrator's note. I've seen metadata that there were people who were sick, were very ill, anecdotal data, I should say, and tested very, very positive for the SARS-CoV-2, for the COVID-19 disease, whatever it was testing for, right? And upon receiving the antibody, the monoclonal antibody therapies, they felt significantly better much faster. Now, having a few anecdotal data points is no proof at all that they wouldn't have just felt better anyways. I have no idea, all right? Presumably, though, those individuals were not given other things, other toxins, and it's possible, as some people have speculated, that there are cloned agents, biological agents, synthetic biological agents, if you will, which cause these COVID tests to be explosively positive and are well neutralized with the monoclonal therapies that were provided by companies such as Regeneron. I'm not really sure what to think anymore about a lot of things. But I will say this much, continuing on with the narrator's note rant here. Here we are entering our 5th year of the pandemic, and what do you want to say? 4 years at least of the 3.5 years of the vaccine availability. And within the United States, at least, authorization for all of the antibody therapies has been rescinded, taken away, nullified, all of them. But the vaccine remains the same, which is odd that the antibody therapies are being taken back because of the supposed mutation of the virus. As if you can speak about the entire collective of a species of something with a singular. It's sort of like saying you have to watch out for the penguin. Be careful not to get bit by the fish. The last thing you want to do is pick a fight with the bear. I mean, it's just odd. You know, it's odd the way they talk about this. But anyways, nonetheless, the virus can mutate, and that doesn't impact the vaccines for some reason. But whatever. All right, continuing on. It may also be the story of how the United States prevents infectious disease broadly in the future. For the first time, HIV vaccines based on Mascola's research are entering humans. And HIV vaccines. Oops. HIV vaccines. Now, this is probably just a typo. They probably meant to say HIV vaccine technologies. But what I see there is HIV vaccines based on Mascola's research are entering humans. Didn't they say coronavirus vaccines based upon Mascola's HIV research or whatever? And while prospects for success remain cloudy, he and his collaborators' work may yet prove key to responding to a broad suite of pathogens and achieving one of virology's new holy grails, a vaccine and a treatment to prevent a coronavirus outbreak from ever occurring. Interesting. So they also admit that that the current vaccines for coronavirus and treatments will not prevent it from happening again. Thought it was a vaccine, but anyways. HIV is Dennis R. Burton, Burton, who's a Scripps Institute researcher commonly regarded as the father of HIV antibody field, said, quote, HIV is the most variable virus out there. So if you can deal with HIV, you can deal with anything. It has pushed researchers to the limit, unquote. Interesting. So HIV is super variable, and it's also a human retrovirus. And exosomes are highly variable also because they are communications. Of course they vary. You don't say the same thing to different people over and over again. You say different things to different people at different times. That's the way communication works. Of course you can't isolate communication. You can't isolate a packet of data on the internet and then have it reproduce. You can't isolate a sound. It is the whole concept of it. You can't isolate individual pieces of constantly changing communication. But they want us to think of it like that. The organized hunt for a broadly neutralizing HIV antibody began in earnest 20 years ago after the International AIDS Vaccine Initiative, a nonprofit founded in 1996 by the Rockefeller Foundation, hired a lanky and bushy-haired ex-professor named Wayne Koff, K-O-F-F, as head of research. Like Moncef Slaoui of Operation Warp Speed, Wayne Koff was tasked with collating and expanding the pipeline of HIV vaccines. Except Koff, K-O-F-F, had millions instead of billions to spend on a problem that, affecting primarily gay men, people in the developing world, and IV drug users, rich nations and companies addressed with far less urgency. By then, the world's hopes for an HIV vaccine had already been deflated repeatedly. When French researchers first isolated the virus in 1983, scientists at Merck and a University of San Francisco spinout called Chiron Corporation, Chiron, were nearing completion of the hepatitis B vaccine. It was a landmark moment in vaccinology, the first vaccine to use recombinant DNA technology. Its success inspired the same enthusiasm that mRNA has today. HIV, it seemed, was just a matter of time. Quote, "We actually thought it was going to be easy to make vaccines for all viral diseases, but then HIV turned out to have quite a few number of tricks up its sleeves," said Dr. Nancy Logan Hagewood. Now, this is a picture from Hagewood in 2011. So one of the first people to work with recombinant DNA vaccines at Chiron Corporation then is one of the most featured scientists in the book Big Shot AIDS Vaccine that comes out the week after September 11th, 2001. All right, I'll buy it. Of course, this book talks about Vical Corporation in a very, very positive way, and VaxGen in a not quite as positive way, but still better than the old school, I suppose. It's very slanderous of the VaxGen executives. She then goes on to make PBS documentaries with Anthony Fauci in 2003 and 4. Yes, I swear to the Lord, she's making PBS documentaries with Anthony Fauci about the HIV vaccine. She then in 2007 is the person that accuses Bruce Ivans of being responsible for the anthrax letters. Research, I should say, an event which, by the way, crushed our Constitution and brought into agencies such as BARDA, the Biomedical Advanced Research Development Authority. She then goes on to become one of the foremost people on the coronavirus response team, again, with a connection to Anthony Fauci there. She's quoted in an article about the future of vaccine and basically all of vaccinology and all of medicine, the greatest achievement in human history, here in this November of 2021 article. Wow. That is quite an incredible set of things. And the company that this other guy, Mascola, just created, Modex, just got a $170 million grant from, guess where? BARDA, an agency made possible by the anthrax letters. I wonder who sent the anthrax letters. Let's argue endlessly about whether or not it was Bruce Ivans. Let's never ask the question of who actually sent them. People may actually start to wonder why Robert Malone and Jill Glasspool Malone and Stephen Hatfill and Meryl Nass, apparently half the people on the Truth and Freedom movement, can't just get together and have a little party and figure out who did it to help us all out. Oh no, can't do that. Let's just all pretend to be good guys and say Ivans probably didn't do it. Anyways, that's pretty freaking remarkable. Virtually all early constructs failed in the lab or in animals, a phenomenon which Wayne Koff was well acquainted. In 1990, as head of vaccine research at the NIH, he hailed how new discoveries, quote, cracked open the door, unquote, to a shot. By 1993, working to build his own vaccine at a biotech, he acknowledged that most of the candidates had captured the world's attention and had failed basic lab tests. Wayne Koff was ostensibly hired to get more candidates into human trials around the world, as the United States would do effectively for COVID-19. But he soon believed that was the wrong task. Narrator's note, why would you hire candidates for a trial when you can simply stage a pandemic and encourage people to take the medicine and then have the government pay all kinds of money out, make it look like it's really the fault of the hospitals and the doctors and get people mad at them. And then you can control the dissidents and the people who are questioning what's going on by having them think that they were figured out the Scooby-Doo mystery and then be mad at all the wrong things, such as the vaccine quality, and silence all the people who are actually asking the real questions. Do you know how many trillions and trillions and trillions of dollars are saved by having people sign up for medical interventions that put their own lives at risk and not have to pay them out should something go wrong, they get injured or killed? You think these companies made a lot of money by selling their products. They probably saved at least $50 to $100 trillion in lawsuits and payouts, which would have had done to people for them to take on the risks that they actually have. That's where the real money happened. And to understand that, you have to accept. You have to look at the underlying causes of elevated death in the United States in 2019-20, 21, and you're going to quickly come to the realization that any increases from a supposed novel, deadly, pathogenic virus are so small that they never would have been noticed if nobody ever even bothered to measure them. First, Wayne Koff, he decided researchers would have to figure out what protection from HIV even looked like, literally. He invited 11 top scientists to a pair of meetings in Amsterdam and New York to hash out a plan. Wayne Koff said, quote, "We realized very, very quickly that HIV vaccine development wasn't a product development problem. It was a discovery problem. If COVID-19 had the envelope protein that HIV does, we still wouldn't have a vaccine today." Interesting. Unquote. A spherical shield around the virus, the envelope is one of HIV's most powerful weapons. To understand just how devious it is, a contrast with COVID-19 is instructive. Antibodies have been the heroes of COVID-19 immunity. Swarming through your bloodstream are 10 billion B cells, each carrying a slightly different antibody on its surface. These amazingly eclectic Y-shaped molecules produced by random shuffling of DNA act first like an army of sentinels. When one latches onto a foreign protein, it starts making copies of itself and spitting out thousands of antibodies per second. Ideally, these antibodies latch onto a part of the virus and interfere with its ability to infect cells, a problem called neutralization. When SARS-CoV-2 enters the body, it makes little attempt to hide its spike protein. Here's my spike, everybody. Spike party. That now famous grappling hook. And for all the concerns about variants, it can't readily change the spike's structure. Within 7 days, most patients are teeming with neutralizing antibodies that can glom onto the spike and prevent it from hijacking cells. One of the reasons most people clear the infection so quickly. Interesting. So, again, playing a little bit and going along with the narrative. If most people clear it out, what's the problem? Why did we have to shut the world down? Why did we have to tell people take it or lose your job? Why did we have to divide families? Why are there still people dying today, 48-year-old fathers of two, who died because they took a medical intervention based upon the perception of risk of the baseline virus? I don't see that to be the win-win. I don't see that to be the fruit of this event. back to the article. Give someone a vaccine encoding for the spike protein, and within 2 weeks, they'll have swarms of neutralizing antibodies coursing through their blood, an army waiting to fire. The RBD, the notorious RBD, is just sitting there like a target that says, "Hit me," says John Philip Moore, an HIV researcher at Cornell University, referring to the receptor binding domain that sits atop the spike. And he said, quote, "an equivalent of the HIV envelope just does not exist," unquote. The HIV's envelope makes sure the virus doesn't fall prey to the same fate. Interesting. It's almost as if HIV, this human retrovirus, is really just exosomal communication. Since it's so hard and so convenient for it to enter cells, it's almost as if cells are designed to use it for cell-to-cell communication, something that every single multicellular organism has, period. Multicellular organisms use extracellular vesicles to be able to communicate with each other, or just simple proteins. That is the way multicellular organisms work. Multicellular organisms did not wait to develop a spine and a nerve system and arteries and a brain and all kinds of other specialized tissues before they could exist. They started off as colonies of single-celled organisms that became more and more optimized to work together as a colony through exosomal communication. This is the basis of multicell organisms. And they're calling it HIV. Oh, it's changing all the time. Well, no, whatever, Sherlock. Of course it's changing all the time. It's sort of like walking into a family and saying, wow, everyone keeps telling everyone else something different all the time. Unless you're being nagged about emptying the dishwasher, in which case that message may have to be sent a few times from one person to another person. You see what I mean, though? The first HIV vaccine ever designed, a construct, Genentech, first put into humans in 1987, elicited plenty of antibodies in volunteers. But not one of them effectively neutralized the virus. Burton said, quote, "the virus always wins," unquote. Well, almost always. Early on, researchers learned from studying the blood of patients that a tiny subset of HIV patients kept the virus in check. And that pointed towards a solution. If someone could figure out what was happening to those patients, if they could isolate one of those antibodies, then maybe they could reverse engineer it. Figure out what it looked like, how it bound to and disrupted the virus, and design vaccines that could induce healthy people to produce replicas. John Maskell, first guy mentioned, and a former Vaccine Research Center president, America's vaccinologist, according to this article, and now chief science officer of MODEX, said, quote, "the first observation was that the natural immune response to HIV in some people, was much better than anything we could get with the vaccine. So then the question was pretty obvious. What's in these people's serum?" Unquote. Fishing individual, sometimes called monoclonal antibodies out of serum, the yellow soup left over when you dump all the blood cells out of the blood, wasn't easy. The original method, pioneered in 1976, invoked taking a B cell from a mouse and fusing it with a cancer cell to create an immortal, antibody-pumping hybrid cell. It was a Nobel Prize. Nobel Prize winning stuff, sparking hopes for drugs of all sorts of diseases. But like many things in medicine, it worked best in mice. Burton says, quote, "and it turned out that it's a lot more difficult to get human monoclonal antibodies," unquote. Burton, who at 69, this again is 2021 article, he'd be 71 now, has a full head of gray-white hair, a quick laugh, and still plays football, the British kind, once per week. Made his name by developing a new approach while on sabbatical at Scripps in 1989. At that time, Richard Lerner's lab, L-E-R-N-E-R lab, was racing with a Cambridge group to isolate the DNA of an antibody and grow it in bacteria. When he arrived from a Swedish university, they were trying to grow just a portion of the antibody, but it wouldn't form. So Burton suggested to the graduate student leading the project that they grow the whole thing. And Burton said, quote, "And unbelievably, when I look back, he listened to me straight off and said, 'Right,' unquote." A few Fridays later, Burton went downstairs to find his photo plates growing black, signifying the lab-grown antibodies were binding to their target. They cracked a bottle of champagne that Burton still kept on his shelf today. It's like it just opened up the whole antibody field, Burton said. It's almost the same story, by the way, that one of the guys of Cesar Milstein and Georges Kohler said. Interesting. You got the champagne. Something happened in the basement near the elevator shaft. I don't think we have a bottle of champagne in our house anywhere. I guess good things you need the champagne before good things happen. Otherwise, you're just whistling in the wind, so to say. Burton says stayed at Scripps, enticed by the science and the San Diego sun. Immediately, he started using his new technology on HIV, believing it held a path to a vaccine. It was an unpopular opinion. At the time, most leaders, 90% he estimates, in the nascent HIV vaccine field focused on T cells. Many doubted that a broadly neutralizing antibody could ever be found. And Burton had trouble just securing monkeys for his study. But in 1994, he isolated an antibody called B12 that neutralized two vastly different strains and protected monkeys from exposure to those specific viruses. It proved his idea was possible, but ultimately the antibody, quote, wasn't all that broad and wasn't all that potent, unquote, Wayne Koff said. Most viruses escaped. The technology was still too primitive. It's almost as if they're realizing that different antibodies can do different things in different people. 1 second, let me let my cat out of the room. Dr. James Earl Crowe Jr., C-R-O-W-E is how you spell the Crowe, said, quote, "I mean, it was really, really difficult," unquote. He also said, "It was a miracle if you can make one antibody in a year," unquote. And he runs the Vanderbilt lab, runs a lab at Vanderbilt, and invented the COVID-19 antibodies AstraZeneca is now developing. Vanderbilt's an interesting college. Lots of secondary connections to Ralph Barrick through there. We'll dig into it some other time. After the meetings in Amsterdam and New York, IAVI, that's IAVI, that's the International AIDS Vaccine Initiative. We've talked about IAVI before. They did a lot of computational immunology work, which involved, of all people, Jay Berzofsky and Murray Gardner, postdoc advisor of Robert Malone. And the NIH established the Neutralizing Antibody Consortium in 2002 to try to accelerate the hunt. They brought together researchers from 4 different labs, a number that eventually grew to 18, combining immunologists, virologists, and structural biologists. They tried to establish new standard procedures for isolating antibodies, testing their neutralizing ability, comparing them to other antibodies, and turning the best ones into vaccines. A rare example of science without ego. The consortium effectively centered around separate efforts at Scripps and the Vaccine Research Center. Scripps, by the way, is very close to Vical Corporation. And I think there was a lot of bleed over from one to the next. In particular, this is where Robert Malone comes in. I'm sorry, I take that back. Scripps is near the, I'm thinking of the Salk Institute. I think Scripps is in the same area, though. I think Scripps, Salk, and Viscal are all in the same area. So I take back what I said a few sentences ago. Mascola, a Bethesda, Maryland native, was deputy chief of the Vaccine Research Center at the time. He arrived in 1999 after a post-med stint at the Walter Reed Army Institute of Research, analyzing the virus and why some of the first vaccines had failed, where he came to some of the same conclusions about the importance of antibodies as Burton did. Burton said of Mascola, quote, "He's very collaborative, not the type to be involved in a dirty race," unquote. The big break started in the year 2006 when Koff and Burton launched, quote, Protocol G, unquote. So few patients made antibodies that neutralized HIV that to find them, scientists would have to look around the world. So over the next 3 years, Koff teamed with local medical centers to collect antibody samples from 1,800 HIV patients across the globe, largely in Africa. They selected for those who had been infected for at least 3 years yet hadn't become sick. The body tends to make the best antibodies after prolonged exposure to the virus because the immune system evolves with the invader, selecting over time for the best defenders, a fact that would eventually throw a wrench in Burton and Mascola's vaccines efforts. All those donors posed a problem. How do you screen them? Only patients whose sera could neutralize numerous disparate HIV strains would be useful. So you would need to match dozens of viruses with thousands of samples. Quote, and that's when we started using robotics, unquote, Burton said. They enlisted a diagnostics company called Monogram to throw hundreds of sera samples against tens of viruses at once. It worked. In an initial study, about 1% of the 1,234 donors screened were identified as elite neutralizers. Something in their blood had outwitted HIV. Koff and Burton didn't have a plan for how they could turn those elite neutralizer samples into antibodies, but the gold rush of antibodies in cancer, powerful new drugs like Herceptin and rituximab, had brought startups with new technologies into the fray. Burton called a biotech in Seattle called Spaltudac, Spaltudaq, Spaltudac, told them they should be working on HIV instead of cancer and offered them $500,000 for each sample they screened. Interesting, because of course, HIV is a virus, and cancer, as was understood at the time, or at least as they were saying it, not a virus, not infectious. But someday you can just turn cancer into something that's a disease that's treatable with a vaccine. Well, then, of course, focus on the HIV and you'll be able to solve the cancer later. What the freak? Spaltudac took 30,000 B cells isolated from one of the neutralizers and turned them into so-called supernatants, putting each into an individual well and stimulating them to pump out pools of antibodies. Testing each pool against a panel of viruses, they found 2 could neutralize most. Back at the Scripps Institute, a prolific new graduate student named Laura Walker, I obviously don't even have a link to her, I don't know who that is, dropped everything and began studying how the 2 antibodies bound to the virus, hoping it could direct the path to a vaccine. The paper appeared in Science in 2009, pointing to the first 2 powerful antibodies against HIV. Laura Walker just ran off with it, said Wayne Koff. The breakthrough began to rekindle interest in HIV antibodies, a shift that accelerated that December with a finding out of Thailand. A vaccine trial there had once again failed, but unlike previous trials, a small subset, 30%, appeared to have been protected for a year, and their serum seemed to contain antibodies. Mohammed Sahadi, Sajadi, who is an HIV researcher at the University of Maryland, said, quote, "Everyone was caught by surprise. And that really opened things up and put the focus back on antibody," unquote. Everything was clicking. In Bethesda, the Vaccine Research Center was also nearing a discovery using a slightly different approach. Rather than separating and analyzing all the B cells from a patient, researchers took those B cells and pooled them together. Then they took one part of the HIV envelope they knew was vulnerable, in many cases a protein that the NIH's structural biologists had identified, and used it as a fishing hook for the best antibodies. In 2010, they found one that could neutralize 90% of the viruses thrown at it. Mascola presented the results to his colleagues in a conference room on the 4th floor, showing a color-coded table representing how well each of several antibodies could neutralize 190 strains of HIV. Green meant it barely bound, but red meant a potential drug. Nearly all of the rows of the antibody called VRC01, that's Vaccine Research Center 01, were bright red. Gary Nabel, the director of the Vaccine Research Center at the time, said, quote, "It was really unprecedented. The first thought that popped into my mind was, wait a minute, do we have it backwards?" Unquote. VRC01, that's the Vaccine Research Center's 01, their first drug, I guess, re-engineered and reshaped the course of HIV vaccines. Longtime skeptics of Burton dove into antibody work. Burton, Mascola, and others identified hundreds more neutralizing antibodies and set about designing vaccines based on the parts of the viruses to which those antibodies. Sounds like that sentence is missing something. Shelley Karuna, who's an HIV researcher at the Fred Hutch Institute, said, quote, "There was tremendous excitement," unquote. Nabel and Mascola, though, wondered if instead of reverse engineering a vaccine, they could just manufacture large doses of the antibody and infuse them into patients. The antibodies wouldn't last forever, and they wouldn't be cheap. Antibodies cost orders of magnitude more per dose to produce than vaccines. But they might help protect people for a while and offer a stopgap until a vaccine becomes available. Let's inspect that paragraph, especially what's in those parentheses. Antibodies cost orders of magnitude more per dose than vaccines. Well, why is that? Because they have to be super absolutely pure. And vaccines don't have to be. That's the answer. There is less quality control in vaccines. There's less quality demands with vaccines, thanks to all of this vaccine-specific legislation. It's not because vaccines somehow are just easier to make chemically. In some senses, they're probably even more difficult, because it's a vaccine is something that you have to engineer. Presumably, it's something that doesn't exist in nature already, whereas the antibodies already exist. You just have to clone them somehow. So guess what? If you can just redefine all disease as vaccine treatable, you can now treat everything with a vaccine. Lower the quality standards, avoid lawsuits, and have an opportunity to exercise the human ribosome to understand how proteins are created based upon a variety of inputs, including epigenetic and DNA. It is a win. Also, because of all of these antibody patent annoyances, you can avoid all of those. You don't have to patent antibodies specifically. You only have to worry about the platform. And you can get all of that by at some point in the future just having people make their own antibodies with transfection, as opposed to injecting them. This article here, I can't believe in retrospect, 2 years later, based upon everything we've learned, how in our face it was 2 years ago that there was this tap dance around this. How do we get people to just use their own bodies to make the proteins directly? Antibodies in this case. How can we get more cash? And how can we get more data? You get a whole lot more data from people. Now you can harvest data from the entire human population. It is so remarkable what has been done here. And people are still talking about the lab leak or the DNA contamination. Or wait, now there's the New Zealand distraction. The distractions are all over the place. However, if you understand the antibody patent paradox and understand the benefits, as I itemized in a recent video, making antibodies directly with transfection in humans, you go back and you will find this in our face in almost every single article over the last 4 years. It's been right there the whole time. And now this company, Modex, this other startup, thanks to Gary Nabel and his connections, hidden connections, by the way, with Vical Corporation, is exercising this. This is. I can just work on my tractor all day and just do like a 5-minute video. We are so freaking far ahead of whatever other channel is talking about. I don't like to be boastful. I don't want to be. I really don't. Because if we are, then we really haven't had the impact that we need to. The point is I just want to do my own thing and listen to other people talk about this. They have mechanisms to get paid, and then I could just, it's just right there. A model of the VRC01 antibody. I guess there's a picture here that I probably didn't download. The strategy has been tried before, most notably with an approved drug to protect at-risk kids from respiratory syncytial virus. Wow. Isn't that amazing? All this talk about RSV. Where the heck did RSV come from? In HIV, it had the potential to not only prevent, but also treat. On the back of each antibody is a beacon that might signal the immune system to clear HIV-infected cells, something that could be essential for an eventual cure. A cure? I thought we were just talking about prevention. I'm so confused. They keep going back and forth. Oh, my goodness. Oh, my goodness. Knock, knock at the door. Knock, knock. Who's there? New people coming to the party. It's David Baltimore and Stanley Plotkin. Oh, Stanley Plotkin, Chief Science Officer of Eros, Chief Science Officer of Inovio, Chief Science. Well, I shouldn't say that. He was tangentially involved with Inovio. Chief Science Officer. No, excuse me. He was the Chief Science Officer of Inovio, the same company that Gary Nabel, Modex, first director of the Vaccine Research Center, by the way, had like a 10, 15-year collaboration with. Stanley Plotkin, who has anthrax vaccine. Oh, my goodness, speaking about anthrax vaccines, work going all the way back to 1960. Stanley Plotkin, who was the Chief Science Officer of CEPI, the Coalition of Epidemic Preparedness and Innovations. This guy's all over the place. So now we have Baltimore and Plotkin at the party. But they decided after meetings with Nobel Prize winner David Baltimore and renowned vaccinologist Stanley Plotkin and other advisors that they needed to at least test the concept. John Mascola said, quote, "We knew that a vaccine could induce these antibodies, was a long way off," unquote. And as the months went by, they gained new reasons for putting the antibody into people. The antibody hunt was supposed to point them to a vaccine, but it quickly pointed to just how hard a vaccine would be. The first constructs Burton, Mascola, and others built based off their prized antibodies failed to produce the same antibodies in animals. Well, of course. Oh, boy. How obvious is that? Broadly neutralizing antibodies for HIV, it turned out, were strange molecules, many of them bound to both the virus and that vibrating glycan forest, something few antibodies do. They had long loops rarely found in other antibodies. Their genetic code contained weird insertions and deletions. In hindsight, the strangeness may not have been surprising. After all, these antibodies were distant outliers, a tiny fraction of the tens of thousands of B cells isolated from just 1% of HIV patients. They were the product of an immune system evolving over years, living with the virus, selecting for the best antibodies over and over and over again as the virus tried to evade each innovation. Narrator's note. Viruses. You all are familiar with the concept of a computer virus, a program that somehow gets into your computer, your electronic device, something that was unintentionally, unknowingly installed. It came in with another program. It came in with something that you might have done on the internet, an email you opened. Okay, a computer virus. And these computer viruses, these programs, once they get on your computer, they are hard to find in the file system. They can run in the background. They can do everything from use your computer's CPU to do a little Bitcoin mining. They can find files and send them out from your computer. They can corrupt your data, or they can just try to get passed on to another computer somehow. Whatever. They can embarrass you. Whatever. There's lots of things that they can do. That's the concept of a computer virus. Computer viruses are designed. Computer viruses are designed by people who write and design and test computer viruses. There are effectively companies. A computer virus is not made by some individual in a garage. They're made by groups of people who work together. They have multiple files. They have multiple machines used for testing and building these products. They have hundreds of machines probably to test them on different versions of different operating systems and different computers. They actually have version control. So they'll have a version 1 of the virus, and then they do lots and lots of testing, lots and lots of code changes, all very intentional, and then they'll make a version 2 of the virus. And that will not get released until it's tested and everything else. The point here is that computer viruses don't evolve. Computer viruses just don't get installed onto a computer and start flipping around their bits and changing their values until they do something different. Computer viruses, every version is very specifically designed and thought out. Computer viruses don't happen by accident. They don't happen because data was corrupted and data changed and now it's something different. They are not random events. This concept that viruses can just change their code and still work and do interesting different things is so flippin' absurd. It is one of the most absurd concepts out there. You did not get Windows 10 because Windows 9 mutated. You got Windows 10 because Windows 9, and I'm not a Windows fan, by the way. but I'm thinking about that now because it's so strange that the computer that's most vulnerable to viruses, so that we always talk about viruses in the spread of computer viruses, is Microsoft. The same company that's connected to all of the genetics and the fetal cell tissue laboratories at the University of Washington and the Bill Gates family, which now is using all of these proceeds for the Bill and Melinda Gates Foundation, which in turn is dumping it all into gene therapy because of the concern of mutating viruses. I speculate that much of the fear of viruses and the pollution of the word viruses into our vocabulary, the fact that viruses exist like they do on Windows, Microsoft Windows, I believe was an intentional act. It was one of the inherent values and the roles of Microsoft was get people to talk and think about viruses and viruses and viruses and viruses and viruses and viruses. Virus, 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 virus spread, infect, virus spread, infect, over and over and over and over again. It's not necessary. With a little bit of quality control as an operating system developer, you don't need to have all these computer viruses. It's as if Windows was designed to provide a platform to evangelize the concept of viruses. No other operating system has these kinds of problems. I'm not saying it's impossible to write a virus or design a virus for another operating system. They all have there's effective ways to do it on every one, but most of them do not offer the opportunities that Microsoft does. Anyways, this whole concept of virus mutation, everyone, look, I mean this figuratively, not literally. But every biologist that just says viruses just mutate and they change and they do different things should seriously be slapped in the face really, really hard by someone who is very, very strong. Repeatedly until they cry and apologize for this. And I'm not condoning violence. I'm actually suggesting it because I want them to be uninjured and so that they can recover and that so they can help the world see how badly they've been bamboozled and turned into bamboozling other people. I had to do that rant. I'm sorry. You see, this article just brings it out in me. But it is meant that HIV researchers would have to invent a new strategy. They would have to figure out a way of coaxing a healthy immune system down that same path these rare patients had followed. Something that no one really knew no one really knew how to do. It says had to do. So typo in the article. Burton said, quote, logically it should work, but we didn't anticipate at the time how long that path would be, unquote. VRC01 would be the best way for a while. And I do believe, by the way, that VRC01 is very much connected with a Vical product. I think that Bob Malone's Vical is very much connected with VRC01, which is amazing. So the whole Vaccine Research Center is based off of a Vical product or using Vical product manufacturing. And didn't Vical just disappear? They finally delisted in October 2019 and were purchased out. And now you have the patent boy as the hero of the resistance and the truth and freedom and transparency and medical whatever movement. VRC01 would soon offer its own disappointments. Mascola set up the Vaccine Research Center's 2,000-liter bioreactor to churn out kilogram after kilogram of antibody, far more material than the center had ever produced. And National Institutes of Health and the U.S. Army labs set up a series of trials in healthy volunteers and then HIV-infected patients. Trevor Crowell, Crowell, hired a new physician at the United States Military HIV Research Program in 2024. Remembered flying out to Thailand for intense 5-day trips to set up a trial there on HIV patients. The study would take volunteers off their medication to test whether VRC01 could keep the virus suppressed on its own. In theory, the trial was blinded and placebo-controlled, but back in Virginia, the results became clear rather quickly. Trevor Crowell got an email every time a volunteer had their virus rebound and had to go back on meds. The updates came in swiftly. Trevor Crowell said, quote, "It was disappointing." Though he noted one patient on VRC01 did keep the virus suppressed for 40 weeks, which was called a glimmer of hope, unquote. A pair of studies from the National Institutes of Health and the AIDS Clinical Trial Group reached the same conclusion. By the time the novel coronavirus rolled around, only one major trial was left, a 4,500-person study to conclusively test whether the antibody could prevent remission. VRC01 may have neutralized 90% of viruses, but practically, with the levels of antibody you could infuse in people, it protected against a far lower percentage of strains worldwide. Catherine Barr, that's B-A-R, single, one R, by the way, said it was an awakening, unquote. And she is a University of Pennsylvania researcher who led 2 of the studies. Even as a vaccine or treatment for HIV receded further into the distance, the neutralizing antibody consortium's work began popping up against other viruses. In 2013, Jason McClellan, a structural biologist who worked with the Vaccine Research Center on identifying how Mascola's antibodies bound to HIV, left the NIH to start his own lab at the University of Texas at Austin. He found out Peter Kwong, Kwong, the chief structure biologist on the HIV project, and Dr. Barney Graham, Mascola's deputy, that's right, Barney Graham was the deputy director of the Vaccine Research Center, to try another spin on the concepts the consortium was struggling to apply. Jason McClellan said, quote, "To me, it was unclear whether these concepts weren't working because they were bad ideas or because HIV was really just a really, really, really hard virus to make a vaccine for. So my idea was, why don't we apply these principles to a more tractable virus," unquote. The obvious case was RSV, the second leading killer of small children in the world after malaria. It is? Do you know anyone who's died of RSV? I'm hearing that there's these massive breakouts in schools and daycares and RSV. You walk into a Walgreens to buy some cashews and, well, RSV, get your RSV shot. Like, well, I don't know. Maybe I don't know. Continuing on, vaccines for RSV were an infamous failure. One candidate in the 1960s actually turned out to enhance the disease. But scientists had found an antibody marked as Synagis, Synagis, that could protect the highest risk children from infection. Protection had to be possible somehow. McClellan took Synagis and a couple other neutralizing antibodies and studied how they bound to the fusion protein, RSV's equivalent of a spike protein. When RSV reaches a cell, it changes the protein's shape for entry. McClellan discovered that the antibodies bound only to the pre-fusion form of the virus, not the post-fusion structure that everyone had been making. With some tricky structural biology techniques, also borrowed from HIV, they swapped some amino acids to create a stable version of the post-fusion protein. Overnight, it became one of the world's leading candidates for RSV, presumably RSV protection. McClellan then used the protein to engineer new antibodies for the virus, including nirsevimab, which AstraZeneca licensed and is now preparing to launch as the new standard of care for RSV. Dr. Crow, I forget his name again, said, I don't think we have to be embarrassed about the progress because it's amazing. And all of these other fields will be benefiting. Gary Nabel and Anthony Fauci proposed applying the same approach to develop a universal vaccine for the flu, another highly variable virus. How do viruses just don't mutate? I'm sorry. That is the they don't. I'm not telling how much about the virus narrative I question. A lot, all right? But even just, let's just give it the possibility that these pathogenic viruses are real. There's no analogy to it. You just don't swap stuff around like that and have it work better. It's so ridiculous. Obviously, no one's ever done assembly programming. You flip around bits and it just doesn't work. It's that simple. A vaccine, let me restart this sentence after a sip. You can see why this takes so long to read this article. Okay, I think we can probably get the rest done within 20 minutes or so. Gary Nabel and Anthony Fauci proposed applying the same approach to develop a universal vaccine for the flu, another highly variable virus. A vaccine roughly based on their blueprint recently showed promising effects in phase 1. For Burton, the isolation of HIV neutralizing antibodies, quote, lays down a marker, unquote, for what's possible. If you can find an antibody to HIV, you can find one to anything. And in 2016, Mascola did, teaming with a Swiss startup called Humabs, Humabs. And by the way, that's the AB is probably a reference to antibodies, that had developed a more advanced form of the NIH's B-cell sorting technology. The agency identified a single neutralizing antibody against Ebola. After the virus returned to the Congo in 2018, it proved to be one of the first 2 effective treatments for the deadly virus, bringing survival rates to nearly 90% when given in early infection. Meanwhile, McClellan started applying what he learned from RSV to a novel coronavirus outbreak in the Middle East called MERS. How convenient. Just in time. He figured out that with a similar amino acid swap, he could make a stable version of the virus's spike protein for the first time. It would be crucial for a vaccine, which Graham wanted to design just in case coronavirus broke out again. Just in case. I mentioned earlier that Mascola, and I'm going to open up a second page here, soft-spoken guy, so obviously that means he's nice, is not upfront about the fact that he did a lot of work with Vical Corporation. Let's go back here to the top, to the V's. U and V, VICAL. And I think that the VRC01 sourced a lot of its property from, or at least at the very least manufacturing capability from Vical Corporation. And Mascola, I should say, Gary Nabel, Gary Nabel has been hiding his collaboration with Vical Corporation, not Mascola. Gary Nabel has, and which goes back to like 1989, 1990, the very start of Vical Corporation, and continued on to the mid-2000s. And noteworthy, though, that Nabel also did a lot of work with Sina Bavari on the Ebola virus, again using the Vical vaccine technology. But here, Vical Corporation, I should have the, I don't have it right in front of me here. I should, though. But Vical also received a grant in 2004, again, for a vaccine for anthrax. But I don't have listed in here. Oh, but you know why? Because these are out of order. There we go. Out of order. I need to fix that. Is that Vical was the first to manufacture a SARS vaccine in the United States, according to the Los Angeles Times via Bloomberg, October 14th, 2003. So Vical was the first company to develop and manufacture a SARS-1 vaccine. And here we have all of these other collaborators, such as Nabel, who works a lot with Mascola. They're pretty tight, actually. If anything, Nabel is a mentor to Mascola. And Barney Graham saying, well, let's just work on a SARS-CoV-2 vaccine, just a MERS one, just in case there's another coronavirus outbreak. So back to the article. When Mascola learned of a new virus circulating in Wuhan, he walked upstairs to a collaborator's office and started planning. This could be the culmination of a life's work, he told Wired. Wow. That's a guy that sounds like he is so busy drinking the champagne. That's a pretty boastful statement for Mr. Quiet, chief vaccinologist of the United States. People are still dying because of the medical interventions. People are still dying. Not just like lonely people that don't matter. Children and parents are still dying and going away from families because of this novel virus narrative. Every day, while they are drunk on the champagne. But it gets better. Since 2009, other companies and researchers had expanded on the techniques they used to develop VRC01. One, a little-known but well-connected Vancouver company called Abcelera, the same company that Brett Weinstein's brother, Eric, told Peter Thiel to double down and invest a lot of money in. Abcelera, that I believe actually sourced its antibodies from the Snohomish County man just across the border in Seattle. Abcelera, the first company that had an antibody design. Of course. It was all set up. We all know it was. Abcelera developed a microfluidic chip that could sort millions of B cells within a day. A microfluidic chip. In 2018, DARPA, the defense agency behind weather satellites and the internet, gave the company a $30 million contract for a pilot project to test whether it could develop an antibody against a virus in 60 days. In mid-January, after the first case in the United States, Snohomish County man, Mascola and Graham called CEO Carl Hansen, who's designed CEO. I mean, probably a great guy, but all those articles about the overnight Canadian billionaire, that's Carl Hansen, previous athlete, guy's ripped, and suggested they ditch the pilot and start on COVID-19. Abcelera had the advanced platform, but it would need Mascola's infectious disease expertise. He immediately agreed, Mascola said of Hansen. Wow. So this company, Modex, their chief science officer, is also the guy, Mascola, who was the middleman getting the antibodies from Snohomish County man to Abcelera. Oh my goodness, and he's working at a company that was founded by Gary Nabel, who's been working with Vical and Sina Bavari and who knows what else since 1989. Something that for some reason didn't make Gary Nabel's Wikipedia page. Calling hospitals and clinicians to obtain the over ethics, Mascola managed to get a blood sample for the... I found it! Oh my goodness! This article actually says that Snohomish County man was the blood sample. Oh my goodness. Woo! Wow. Snohomish County man was the antibodies. Wow. We looked at 100 and only one was good. Okay, so it doesn't say that specifically Snohomish County man was the one of the 100s. But I know from elsewhere that of the first 20 people that were identified, I have this quoted elsewhere, only one of them agreed. And I forget how I did the math. But yeah, I triangulated it down to it must have been Snohomish County man. And Don Mascola was that guy. Holy crap. Wow. I just keep rereading the old articles. And with retrospect, guys, I don't have a chat window open. I'm sorry. I'm missing some of the best stuff right now. I just keep reading the old articles with the new insights. And I can't believe how much is there. Wow. Wow. Future samples from other donors would prove to be more potent, but they didn't have those then. Of course not. All they needed was Snohomish County man. And isn't it crazy that the first vaccine designed, at least within the United States that I know of, for COVID, was that Icosavac stuff, which came out of University of Washington, based on, guess what, Snohomish County man. And all the modeling of the spike protein worldwide came out of modeling out of the Wiesler lab, also University of Washington, based on Snohomish County man. And because of some confusion, by the way, some confusion regarding the viral origin, it is actually possible that Snohomish County man, WAO1, is the root of all the antibodies and is the root of all the models of the spike protein and is the root of all the designs for all the vaccines. One guy, unphotographed, never named, 35 years old, from Wuhan on a flight that doesn't exist. Didn't get anyone else sick along the way. Surviving only because of remdesivir, which was given express emergency approval by Sina Bavari. Oh, good news, everyone, by the way. Today, the book on the RFK Jr. book on Wuhan goes live. And guess what? Housatonic ITS is referenced. How many times? Including, I believe, a mention that Sina Bavari at the end of 2018, early 2019, along with Alison Totura, last postgrad student, Ralph Barrick, wrote a paper about how remdesivir, GS5734, is the most likely thing to work as a therapy, late-stage therapy and antiviral, against a novel coronavirus, very SARS-like, causing a pandemic that comes out of China after possibly leaping off of an animal. I'm going to wrap it up here. I'm not going to finish reading the article. There's still like 10 more paragraphs to go. I'll read this and though. Sequencing data from infected patients showed VRCO1 protected against a subset of strains, but it was going to take broader and more antibodies to prevent infection. Researchers are now testing those, presuming talking about HIV again. Testing those, bringing a combination of antibodies into the clinic in hopes of hitting two spots in the virus that will make it harder for HIV to evade. Nabel, who left the NIH to be The chief science officer of Sanofi from 2012 to 2018 even developed a tri-specific antibody to hit 3 spots. He is now developing that as part of his new company, Modex. But what they don't say here in this article in 2021, that they will now say in 2023, because now it's safe, the coast is clear, their plan is not to manufacture these tri-specific antibodies directly. They're not going to have to deal with all the antibody patent issues. Instead, they're going to have people make their own freaking tri-specific antibodies through the marvel of transfection. Because transfection, you can call it a vaccine. You can avoid the lawsuits, you can get all the money. You can avoid the patent issues, and in the process, if people die, oh well, hey, we're protected, it's a vaccine. It's not a therapy anymore, it's a vaccine. And through all of this surveillance that's being installed, and the threat of ever more and more pandemics and more diseases and more sampling, with more and more use of these therapies, you can get more and more data from people. Because that's the goal. Not just get everyone's DNA. That's nothing. They need everyone's DNA every day based upon who they contacted, what they ate, what they were injected with, where they went, the air they breathed. And they need to put as much of that data as possible across as genetically diverse set of population as possible for as long as possible into the big data analytic computers and try to find patterns. Hopefully try to find patterns. That is what all this stuff is about. They don't want to monitor you. They don't care if you live or die. They don't want to monitor you. Monitoring people is a pain in the neck. All that infrastructure, all that code. You want to spend your time at the beach. All that surveillance is a pain. It's being done for a reason, and it's not to keep people in line, and it's not to make people unhappy and oppressed and kill them and maintain powers and increase taxes and all that other stuff, or because Satan wants to do it, or blah, 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 blah. It's done for data collection, and that's it. And how do you get people? How do you get people to live the life of a test monkey in a cage with limited access to other monkeys, with controlled and monitored food from monitored food sources, testing them every day and getting them to give up their fundamental rights? You know what you do? You trick them into it. Tell them there's never-ending pandemics. And then you get them to think that all of this surveillance stuff is because of terrorists or because Satanists are running the government and they just want to make you miserable. You get people to live the life of a test monkey without them getting any reward for it. You get them to forfeit their freedoms, their access to any retribution or compensation should there be any injuries. You deprive them of the ability to know what the real risk reward is of everything that they are being exposed to through phony narratives on all sides of the equation. That's what you do. In closing, this is a fantastic article. How much is said? I can't believe it. And I'm just going to keep reading it again. Like every year, I'm just going to read it again. There's several other articles that fall into this category. It explains why everyone is there in the alt-freedom, controlled, whatever mafia, as well as the mainstream. It describes why things were done. You can see the roadmap here being laid down. Wow. And I'm so glad that we have just stuck with it. And we're not getting distracted by all the other noise out there. So with that said, I am now going to go back and finish changing the oil in my tractor. One of the wonderful things about what I'm doing is I'm just ignoring all the other noise. So it's so wonderful. It's impossible to distract me. All right, guys. Thank you. Take care. Love you all. God bless.